Okay, everybody, welcome to the video podcast for the lab covering the muscular system. This is simply a list of the regions that you'll need to know. You'll need to know the names of these muscles, their location, and what they do. So make sure to review this material and be able to provide this information on a lab practical or on a lecture test. Okay, so muscles of the head and neck region as listed, the muscles of the chest, shoulder, and back region as listed. These are the muscles that you'll need to know for the abdominal region. Know both the muscle and the action that it performs, and the muscles of the hip, thigh, and leg. Again, know the name, the location, and what they do for each of the muscles listed in this group. So what you're looking at here is a little rundown of how muscle action works and some of the important terms that we need to understand when we discuss muscles. And these include the origin, the insertion, and the action. And essentially, skeletal muscle is designed to produce movement. And it does this by crossing a joint. A joint is where two bones come together and are able, through the action of muscle, to perform a type of activity, an action, a movement of some type. Typically, the bulk of the skeletal muscle that moves a joint lies proximal to the joint that it crosses, meaning nearer to the trunk. All skeletal muscles have at least two attachments. One is the origin, which is the attachment to the immovable part of the joint, the bone that does not move, and the insertion, which is the attachment to the part of the joint on the bone that does move. Skeletal muscles can only pull, they never push, and this is because their primary mode of action is contractility. During contraction, a skeletal muscle insertion moves towards the origin, so as the muscle shortens, the origin and insertion draw closer together. And you can see an example here using a muscle called the brachialis, which lies just underneath the biceps brachii in the upper arm, the brachial region. You can see here that the origin is midway down the humerus, just a little bit medial to the deltoid tuberosity, which remember lies on the lateral aspect of the humerus, about a little bit less than midway down the shaft, and the insertion is here on the ulna, okay, on this little roughened surface here, again called a tuberosity. This is a tuberosity that lies on the lateral surface of the ulna. This is the part of the joint that's going to move when the action is performed. So when the brachialis contracts, what it does is it causes the angle between the radius and ulna and the humerus to shorten, and this is what we call flexion. Now, the tissue that attaches the muscle to the bone is called tendon, and the tissue that attaches bone to bone and stabilizes the joint, these are called ligaments. Okay, so you can see here again the origin, the insertion, the muscle name, the action, which is flexion of the elbow, and we're going to use this as a way to identify the muscles on the subsequent slides that we're going to look at here. So in the head and neck region, we see a lateral view here of the superficial aspect of the neck and the head, the cervical and the cephalic region here. First muscle we want to look at is the frontalis. This is the frontalis here, and it raises the eyebrows and wrinkles the skin of the forehead. Okay, its origin is the epicranial aponeurosis that runs across the top of the cranial cavity, and it inserts just above the eyebrows, and it allows you to raise the eyebrows. The orbicularis oculi is this circular muscle here around the eye, and it closes the eyelids. And whenever you see orbicularis, think orbit, think circular path. If you look at this muscle, it seems to form a ring around the eye, The orbicularis oris is around the oral cavity, and it opens and protrudes the lips. Okay, so this opens and closes the eye. This opens and protrudes the lips. The zygomaticus, shown here, runs from the zygomatic bone, the cheekbone, down to the corner of the mouth, and it raises the corner of the mouth, so this is one of the muscles that's active when we smile. The buccinator compresses the cheek, and it's the muscle that's used in sucking and blowing. Okay, and you can see that here. It lies underneath the zygomaticus and also underneath this muscle here, which is called the masseter. The masseter runs from the mandible and inserts onto the inferior border of the zygomatic and the temporal bone around this ridge that forms the cheek, and it's the main muscle involved in chewing, and those are the lateral muscles. Now if we move around to the other side of the model, this is the deep aspect, and the way that you can tell is that a lot of the muscles that we were able to see on the right side of the model we can't see on the left side, and the idea here is to give you a look at the muscles that are close to the surface as well as the muscles that are deep underneath. So here you can see just another view of the orbicularis oris, which opens, closes, and protrudes the lips. Okay, so to review, orbicularis oris, a few other muscles that you don't need to know, but just by way of showing you where they are, this is the temporalis. Okay, the muscle here is the corrugator supercilii, and this muscle that we'll talk about in a moment, this is the sternocleidomastoid. Okay, so named because it inserts onto the mastoid process of the temporal bone behind the ear, the clavicle, and the sternum down the anterior aspect, and it moves the head. Okay, so these are just a few of the actions that are possible at the joint that's formed between the head and the chest. Okay, you can see here flexion, which is the movement of the chin onto the chest; extension, which is raising the head up to anatomical position; and hyperextension, which is looking straight up, and as a result moving the head backward. And so the muscles involved in flexion include the sternocleidomastoid, among others. The trapezius is involved in extension, particularly the upper border of the trapezius, known as the cervical head. And then hyperextension, also facilitated by the trapezius, as well as a few other muscles that lie underneath it. The sternocleidomastoid is shown here, and it flexes the head onto the chest, and it rotates the head. And again, it inserts onto the mastoid process of the temporal bone behind the ear, the clavicle, basically the collarbone, and the upper portion of the sternum. So it flexes the head onto the chest, and it rotates the head if only one or the other sternocleidomastoid contract. This is a paired muscle. There's one on the left-hand side of the head and one on the right-hand side. The trapezius, which is, you can see a little bit of back here, okay, it's a big diamond-shaped muscle in the back, elevates, depresses, and adducts the scapula, and it extends the head. And we'll see on the models in the lab exactly where the trapezius is located, and you'll understand the action that it performs. Here you can just see another look at the sternocleidomastoid. This is its point of attachment to the clavicle, or I'm sorry, to the sternum. You can see here where it attaches essentially to the upper part of the sternum, called the manubrium, and you can also see the sternocleidomastoid's attachment to the clavicle. Okay, there's a clavicle, and there's a clavicle, and there's another head of the sternocleidomastoid. And so you can see that when this muscle contracts, it's able to move the head onto the chest by pulling it forward. This is just a look at the back of the model. And to orient you, the sternocleidomastoid is visible over here. We can't see most of it because most of it is lateral or anterior. We're looking at a posterior view of the muscle model here, but you can see here the trapezius. Again, this is the superficial side of the model, and this is the deep side of the model. Okay, so this right here, this whole muscle that I'm tracing, this is one trapezius muscle. There would be another over here if we were also showing the superficial side. So you can see here the different heads of the scapula, the clavicular, the uh, the lower head here that inserts onto the end of the scapular spine and onto the thoracic vertebrae, and the middle head as well, which inserts just below the cervical vertebrae and onto the superior border of the scapular spine. So 1, 2, 3 heads, all that do slightly different stuff. So this head elevates the scapula and extends the head. This it adducts the scapula, basically brings it back towards midline, so the scapula would rotate like this. Okay, I'll draw an arrow so you can see. This will cause the scapula to rotate like this. Okay, and this depresses the scapula, so this pulls the scapula down. Okay, and this right here would elevate the scapula and would extend the head, that is to pull the back of the head back so that you're looking straight up. Okay, so those are the actions. And the trapezius is a very large muscle located on the superficial upper back, and it runs again from the scapular spine all the way to the vertebrae, and it runs from the cervical through the thoracic and down into the upper lumbar region. This is just another look at the same model. You can see here the sternocleidomastoid from the lateral aspect, and that's the sternocleidomastoid right there, and the trapezius from the lateral aspect here. Okay, the trapezius is going to be on more towards the posterior side. Okay, this should be a B right here. Let me correct that real quick. Okay. Okay. So sternocleidomastoid and trapezius. Okay. Some other motions that are possible, and we're looking here at the muscles that move the upper and lower limbs. You can see here an example of flexion of the arm. Okay. And again, this is reducing the angle between the arm and the plane of the body. Okay, so what we're doing here is bringing the arm up. Here we're looking at flexion of the knee, so we're reducing the angle between the fibula and tibia and the femur. Okay, here you can see extension, bringing the knee forward, and extension of the arm, bringing the arm back. Okay, remember that the arm is the upper arm here, referred to as the brachial region, as opposed to the forearm, which is the antebrachial region down here, distal to the elbow. Okay, what you're looking at here are the muscles of the trunk, some of the major muscles. And if you remember your body regions back from the chapter where we talked about areas of the body and what they're called, such as the cephalic region is the head, the cervical region is the neck, the thoracic region is the chest, and so on. You can refer to that diagram, and the names of some of those regions are going to show up in a lot of the names that the muscles have that we're going to talk about. So the pectoralis major is this big broad muscle here in the front of the chest. Now remember, when you're looking at these muscle these muscle models, that the left half of this picture is showing you what's near the surface. This is the superficial aspect, And the right half of this picture is the deep aspect. Okay, so these are the muscles that are closer to the surface. These are the muscles that are under the surface down here. And the pectoralis major is this superficial muscle. It flexes, adducts, and medially rotates the humerus. Okay, so when you think of flexing the humerus, remember that you're talking about decreasing the angle between the humerus and the anatomical plane. Remember, everything is relative to anatomical position. And so what you're doing essentially with the pec by flexing is you're raising the upper arm. Okay, that's flexing the humerus. Medially rotating it is turning the lateral side of the arm inwards towards the chest. Okay, so medial rotation. And then adduct means to bring the arm back towards midline if it's away from midline. Okay, so adduct the arm. The latissimus dorsi is not visible because it's a big diamond-shaped muscle on the back, and we'll see that muscle. Actually, we saw that muscle 2 slides ago when we were looking at the muscles of the back. I think right here. Okay, this is the latissimus dorsi right here. Okay, also known as your lats. So this is the superficial side of the model, and the broad muscles of the back are often well developed in people who do stuff like swimming a lot. So you can see here the lats. These are the traps. Okay, the trapezius, and then you can also see here a little bit of the deltoid muscle. Okay, so we're talking here about the lats. The lats are a little tough to see from this side because they're behind, okay, but you can see here the pectoralis major and the deltoid. Okay, this is the deltoid muscle. It gets that name because it's shaped like the Greek letter delta, which looks like a triangle. The deltoid abducts and flexes and extends the arm. Okay, so flexing the arm, that's raising the forearm. Okay, abducting the arm, that would be moving the arm, and I'll draw the directions here with a pen. Let's do flex first. Flex would be to lift the arm upwards in this direction. Okay, to abduct the arm would be to lift the arm like this. Okay, and to extend the arm would be to bring it out again behind you. Okay, so instead of lifting the arm up, it would be to turn the arm backwards. So we'd have the arrow going into the screen in this direction. Okay, so those are the motions that the deltoid provides because it's got 3 parts to it. It's got a part that's in the front, a part that's in the middle, and a part that's in the back, and they all are part of different motions. The lats will extend, adduct, and medially rotate the arm, and again, we saw the lats in that previous slide. This is just a look at the different kinds of movement that we're talking about so that they're clear in your head. You can see here abduction, right, carrying the arm away from midline, and adduction, carrying the arm back towards midline. You can do the same with your leg, okay? So abduction to carry away, adduction to carry back towards. The delts involved in abduction, the lats and the pecs involved in adduction. Here we've got circumdiction. This is tracing a circle with the extended arm, okay, with the tips of your fingers while keeping your wrist and your elbow stiff. Okay, so that's what circumdiction is. And you can see that movement here. And then you've got rotation, okay? A lateral rotation is turning the head to one side so that you're facing your right or your left. And medial rotation is turning your head from one side to facing straight ahead, such as you see in anatomical position. Okay, so lateral rotation, medial rotation, okay, and lateral rotation again to the other side. And the muscle that does this is the sternocleidomastoid, which gets its name from its attachment points. It attaches to the sternum, the breastbone, the clavicle, that's where the cleido comes in, which is your collarbone, and the mastoid process, which is the little bump behind your ear that's attached to the temporal bone. And then down here, you've got rotation of the arm, okay, medial rotation, again accomplished by the pectoralis major and the latissimus dorsi. Again, that would be if you're standing in anatomical position and you turn your thumbs from facing laterally to facing towards the midline of your body so that the back of your hand is facing forward. Okay, that's medial rotation, sometimes also called pronation. Okay, and then you can see here an example with the leg, medial rotation, turning the foot from facing outward laterally to facing towards midline, and then lateral rotation, moving the foot so that it faces laterally. Okay, so you can do this with the arm, with the leg, with the head. Okay, all of those have rotational capability. You can also do it with the trunk. Okay, you can laterally and medially rotate the trunk. And so this is just a look showing you where the latissimus dorsi is. Remember, this is the superficial side of the model. This is the deep side of the model. And those are your lats, or latissimus dorsi. And there's your deltoid, okay, the triangular shaped muscle that allows you to lift the arm away from midline. Okay, so you want to not only know where these muscles are and what they look like and what they're called, you also want to know what they do. Over here, you've got some major muscles in the brachial region. Okay, the brachial region, remember, is the arm. The biceps brachii, shown here, okay, is called the biceps because it has two heads. Okay, bi meaning two, biceps, two heads. Brachii means in the region of the arm. And what does it do? It flexes and supinates the forearm, and it flexes the arm. Okay, so if we were to define those, flex and supinate the forearm. Well, flex the forearm means what? It means to basically bring your forearm, to bend your elbow, essentially, from anatomical position. To supinate the forearm, you would stand in anatomical position with your palms facing forward, and then you would turn it so the back of your hand faced forward. Okay, that would be supination. And then the triceps brachii is a three-headed muscle. That's why it's called tri. And it's on the back part of the brachial region, the back part of the arm. And what it does is the opposite of the bicep. It extends the forearm, so it allows you to unbend the elbow, okay, to extend the elbow. And it also extends the arm, okay, so it's partially responsible for bringing the arm back behind the anatomical plane if you're in anatomical position. And we'll see those movements here in just a minute when we look at the actions of these different muscles. Okay, so the biceps brachii, the triceps brachii. Again, bi meaning two heads, tri meaning three heads, brachii meaning in the region of the arm. Okay, so here you can see these different movements. Okay, there's the biceps brachii involved in arm flexion. Okay, and you can see here the triceps brachii involved in extension of the arm, bringing the arm behind the anatomical plane. Okay, so these two muscles are antagonists of each other. They basically do the opposite of what the other muscle does, and as a result, they're responsible for controlled movement of the arm. Okay, here you can see a model of the arm, and you can see these major muscles. You can see here the biceps brachii. Okay, again, this is the deltoid, this is the biceps brachii, and down here this is the triceps brachii. And you can also see the triceps brachii here. We can't see the biceps brachii from this view because we're looking at the back of the model. We'd have to be in front of him in order to pick up the biceps. Okay, if you think about the biceps, think about the cartoon Mighty Mouse or Popeye the Sailor when they would make a muscle. Okay, that would traditionally be the action of the bicep. Okay, and here we can see a look at some of the other movements that the biceps aids in. The biceps flexes and it also supinates the forearm. And remember that that's standing in anatomical position with your palms facing forward and your arms at your side. Okay, that is the supine position. The prone position would be with the back of your hand facing forward. Okay, so what the biceps does it can supinate the forearm, so it can make it so that you go from the back of your hand facing forward to the front of your hand facing forward. Okay, so that's supination, that's pronation. Okay, you can see here that in the supine position, the radius and the ulna are not crossed over each other, whereas in the prone position, they are. Okay, and that's all because of the action in part of the biceps brachii. And here you've got to look at some of the muscles of the abdominal region. These are some very big, broad, basically flat muscles that help hold your internal organs inside your abdominal pelvic cavity. And they're also important in certain movements of the leg and the trunk. Okay, remember that the thigh and the trunk. Remember, the thigh is the upper leg, and the trunk here is essentially what I'm tracing now with the arrow, with the cursor. So what do these muscles do? They flex the vertebral column, so they can bend the trunk forward like you're doing sit-ups, okay? And they can turn the vertebral column from one side or the other, okay? So they can be involved in lateral and medial rotation, and they also compress the abdomen, okay? These are the muscles, again, that you use when you do your sit-ups. And those muscles are the external obliques, the internal obliques, and the transverse and rectus abdominis. Okay, so in this group of muscle names, like we can start with the easy part, right, which is abdominal, meaning what? Meaning that it's found in the region below the diaphragm and above the hip. Okay, that's your abdominal region. And these names here at the end of the muscle describe, here oblique describes the direction that the fibers of the muscle run. Okay, oblique means runs at an angle to the long axis of the body. The fibers in the external oblique muscle, they run from basically superior to inferior towards midline. If you want to think of the way these fibers run in the external oblique, think about when you put your hands in your pockets, okay? That's a way to remember which direction the external oblique fibers run. The internal obliques run at a right angle to those muscles, and the internal obliques are just underneath the external obliques on the lateral side of the abdominal pelvic region, and they're also important in moving the trunk and in flexing the hip. Okay, so finally we have another set of muscles down here. Again, abdominis, the transverse abdominis. Abdominis means in the region between the diaphragm and above the pelvis. And transverse means that the fibers run at a right angle to the long axis of the body. Okay, so the transverse abdominis muscles are shown, trying to find, they would be found at midline on the back side of this model. Okay, they're a little tough to see here in the front. Okay, they're easier to see in the next slide that we'll take a peek at in just a minute, because this is a deep set of muscles that really runs underneath this set of muscles shown here. Okay, indicated by D. These are the rectus abdominis muscles, and what they do is they run along the long axis of the body. Okay, so we should probably do a little bit of relabeling here, so hold on just 1 minute. Okay, we'll save that. Okay, and so now you can see, okay, the rectus abdominis, the internal obliques, the external obliques, okay? And then if we come around to the other side of the abdominal pelvic cavity, you can see here the transverse abdominis, and see how the fibers run at a right angle to the long axis of the body. This would be the long axis of the body. This would be at a right angle to it, okay? So the name is telling you the direction of the fibers. Now, something to keep in mind is that not every muscle name is going to have every single descriptor in it. Some are going to have just a reference to the region of the body and perhaps the shape. Some are just going to have a reference to the direction of the fibers and the shape, or some might be a descriptor of the relative size of the muscle in a muscle group. So it's not a hard and fast rule that every muscle name will contain every feature of the muscle. But what you can do looking at these names is you can pick out some important descriptors in the name that are going to tell you generally where the muscle is located. Okay, here you can see the hip, thigh, and leg region, and a couple of muscles that we want to be aware of here are the psoas major, okay, which is this muscle right here, and the iliacus muscle, which is this muscle here, and they're sometimes referred to collectively as one muscle called the iliapsoas, okay, so that would be this entire thing right here, okay, and this muscle is involved in flexion of the hip. It also flexes the trunk and the thigh. And again, what's in a name? In this case, the iliacus is telling you that it's attached to that portion of the hip bone known as the ilium. That's this big crest up here that forms the fan-shaped part of the coxal bone. In the psoas major is just telling you the relative size of this muscle in comparison to other muscles nearby that we really can't see because they're sort of buried. But this is the largest muscle in the collection of the psoas muscles, which are essentially located just lateral to the midline of the body and extend all the way down from the lumbar spine to the femur. Okay, you can see the muscle running down through here. This is a look at the gluteal group. Okay, so the gluteal group is actually a group of 3 muscles, and going from the most superficial muscle to the deepest muscle, what we have on the very outside is the gluteus maximus. Okay, these are the muscles you think of when you think of your posterior. Okay, it's the most superficial and the largest muscle in the group. It extends the thigh. It helps us walk. Okay, another muscle that's in this same group that lies right underneath the gluteus maximus is the gluteus medius. Okay, it's a slightly smaller muscle just underneath the maximus, and it is involved in abducting the thigh. So basically, from anatomical position, if you were to raise one of your legs out to the side, that would be thigh abduction with your knee straight. Okay, and then there's another muscle that's underneath the medius, which is called the gluteus minimus, and it's the smallest muscle in the group, and it basically assists the gluteus medius with its desired action. Okay, so there you go with the gluteal muscles. Now, here we can see the gluteus maximus removed, and we can see underneath, and we can see the entire gluteus medius. Okay, you can see it all through here, the muscle that lies just underneath the maximus. Okay, so we've pulled that muscle away on this side of the model, and you can see underneath. The minimus would be right underneath this muscle. Okay, it's a little bit tough to see, but there it is. Next thing we want to do. Okay, what we're looking at here is just the medial region of the leg. So we're looking basically from the inner thigh and calf towards the lateral view. Okay, so the muscles visible include the gracilis, this strap-like muscle here that's important in the adduction of the leg, so it brings the leg back towards midline. We can also see up here the sartorius, which is an S-shaped muscle that runs across the surface of the anterior thigh and inserts just underneath the patella onto the tibial tuberosity. And this is responsible for flexing the thigh and the leg and rotating the thigh. It rotates the thigh laterally. And we can also see the adductors, this group of muscles here, indicated by A. And what they do is they attach from the pubis to the medial region of the femur, the medial border of the femur, and they help to adduct the leg, bring it back towards midline as well. Other muscles that we need to be aware of that are a little bit tough to see include the tibialis anterior, which runs just lateral to your shin bone, okay, the tibia. Again, the name is telling you the location of the muscle, tibialis anterior, anterior meaning the front, tibialis meaning near the tibia, and it's responsible for raising the toes. Okay, so it is a toe lifter. That action is called dorsiflexion. Okay, and I'll draw here what it is that it makes the foot do when it pulls it up like that. Okay, and we also want to be aware of the calcaneal tendon. That's this area here. It's attached to two muscles that are very important. The gastrocnemius, okay, which is responsible for pointing the toe, so its movement is to point the toe down. This is how you're able to push off on the ball of your feet when you walk. And there's also another muscle underneath it you can see right there. That's the soleus, and it does basically the same movement. A couple of things that we want to be aware of when we're talking about the leg is that the sole of the foot is what we call the plantar surface, and the top of the foot is what we call the dorsum. Okay, so the tibialis anterior is important in dorsiflexion, and the gastrocnemius and the soleus are important in plantar flexion. Okay, two movements that are the opposite of each other. And then if we move on here, what we're looking at now is the anterior thigh and calf. Okay, so this is defined as the femoral region, and this is defined as the crural region, C-R-U-R-A-L. And we can see here a group of muscles across the front of the thigh that are called the quadriceps. Quadriceps right here means fore, and femoris means near the femur. The femur is the big, long, strong bone in the upper leg. It is the longest and strongest bone in the body, and the quadriceps are there to extend the leg and to flex the thigh. Okay, you can see some of the adductors here. If you look in this close-up view, again, they run from the pubis to the medial border of the femur, and what they help do is they help pull the leg back towards midline. So if we were to draw that, it would be this kind of motion here, okay, drawing the leg back towards midline. You can see the sartorius here, okay, this strap-like muscle that runs from lateral to medial. Again, it's shaped like an S, runs across the front of the thigh, and it is again responsible for lateral rotation of the leg and rotating the thigh. And the tibialis anterior, which is right here, okay, this would be the medial side of the leg, and this is the lateral side of the leg. And what the tibialis anterior does is it engages in dorsiflexion, so it lifts the toes up, okay? So those are the movements, and those are the related muscles. The reason that the quadriceps femoris are called the quadriceps is because there's actually 4 muscles in the quadriceps. You don't have to know these 4 muscles, but I'll show you where they are. Okay, they include the vastus lateralis, the rectus femoris, and the vastus medialis, and then under the rectus femoris, a 4th muscle, okay, which is hidden from view here, called the vastus intermedius, and that's the 4th of the 4 quadriceps muscles. And what do? Again, they make it possible for you to kick, okay? Those are your kickers. Now, if we look on the back side of the leg, we see a different group of muscles here. These are the gluteal muscles, okay? The gluteus maximus is responsible for extending the thigh and helping us to stand bipedally. And then down here, you've got the hamstring group. The hamstring group is made up of the biceps femoris and the semimembranosus and the semitendinosus, the 3 muscles of the hamstring group. And what they do is they help to bend the knee, okay? So that's what we call knee flexion, The gluteus medius is a muscle that you can't really see here because it's actually underneath the gluteus maximus muscle, and it's important in abducting the thigh, basically moving the thigh in this direction away from midline, okay? That's what the medius would do. The gastrocnemius you can see right here, okay? It's this muscle, and it's responsible for pointing the toe, This is the muscle that helps you walk, and it is attached to the calcaneus, the heel bone, which is right there, by a tendon, which is called the Achilles tendon. Okay, you may have heard of people having a ruptured Achilles tendon, and if that happens when they lift their leg up, their foot simply flops and they're unable to move. Okay, and so you can see here again, that is the gluteus maximus. Okay, these are the hamstrings. Okay, I'll check them off there. There's 3 muscles in the hamstring group. This muscle's on top, this muscle is underneath. I'm checking it twice to show you that it comes out underneath this superficial muscle here. And then you can see down here, this is the gastrocnemius. Okay. Okay. Underneath the gluteus maximus is the gluteus medius. Okay. And so it's not visible in this picture because the gluteus maximus is covering it. And here you can see an example of different kinds of movement that are possible with these different muscles. Here we see knee flexion, and the muscles that act here are the hamstring group and the gastrocnemius to some extent. Okay, remember the gastrocnemius is down here, and the hamstrings are back here on the back of the thigh. And then extending the knee is the quadriceps, the 4 muscles that are on the front of the thigh that are responsible for extending the knee, and the movement is shown here. Okay, so flexion, extension. Okay, leg movement shown there. We want to be aware of the 3 types of muscle and where they're found and what they look like and what they do. This is table 6.1 in your textbook, so make sure and star this chart and know this material. You want to understand what they look like under a microscope, and you want to understand the characteristics of all 3 cell types. And so what you're looking at here as we go down column by column, skeletal muscle, which are the muscles that we've been talking about up to this point, is both striated and under voluntary control, meaning we tell it what to do. Striated because if you look real close, you can't really see it here, but if you look in your book at this table, you'll see light and dark bands running vertically along these muscle fibers. These are muscle fibers here. Let's indicate them. Okay, that is a muscle fiber. Okay, that is a muscle fiber. Okay, and these are called myofibrils. Okay, that is a muscle fiber, and so on. Okay, you can see these different muscle fibers. Okay, and what these skeletal muscles are basically very long cylindrical cells that contain lots and lots of muscle protein. The main proteins are actin and myosin, and they're arranged in such a way that they periodically overlap each other, and that's what produces the dark bands, and the areas where they don't overlap is what produces the light bands. And the way that muscle contracts is that these light and dark these proteins that basically interdigitate just like cogs on a wheel, okay, they will walk over the top of each other using ATP as an energy source, and what this will do is allow the muscle cell to shorten, okay, and this is what produces contraction. You can also see connective tissue between the muscle cells. We'll see in a minute how the connective tissue in skeletal muscle is arranged. You can see it a little bit here. The outer covering is known as the paramecium, and then deep to that is the epimecium, and then deep to that is the endomysium. Okay, and what does the connective tissue do? It is the body's glue. It helps hold the muscle cells together so that when the muscle contracts, it doesn't come apart. Um, and here you can see a chart showing you the speed of contraction. Here you can see that it can vary from slow to very fast. So that's slow, that's faster, and that's fastest. Um, by comparison, if we look at cardiac muscle, it's different from skeletal muscle in that there's only one nucleus per cell. This muscle is only found in the heart. It is striated and it is under involuntary control, which means that it's governed really by two things. It's governed by the autonomic nervous system, and it's also governed by the wiring inside the heart, which is called the cardiac conduction system. And these little dark staining areas here, these vertical lines, are a type of feature in the cardiac muscle that's called intercalated discs. And what they do is two things for the muscle. They hold the cardiac muscle cells together, and there's also little tunnels in these discs where the heart muscle cells come together. And these tunnels allow electrically charged particles to move very quickly from one heart muscle cell to the next. And that's important because that helps electrical signals spread very quickly across the heart muscle wall. And that's one of the reasons why the heart is able to contract in response to that electrical signal as a unified organ. Okay, the heart has four chambers. The upper two are the atria, the lower two are the ventricles. And when the heart squeezes, the upper two chambers squeeze first, and then the lower two chambers squeeze. And when they squeeze, they shoot the blood to the body and to the lungs simultaneously. Okay, and that's only possible because the heart muscle is capable of rhythmic contractions that are unified, that are coordinated. Okay, there are connective tissue components in the muscle cell as well. There is an endomysium that's attached to a sort of a fibrous skeleton that supports the heart. And we can also see striations, okay, periodic light and dark bands. Heart muscle cells are highly branched and they have one nucleus per cell. So that's another difference between them and skeletal muscle, in addition to the fact that they're not under voluntary control. We can see that the speed of contraction is slow in heart muscle tissue, and that's so that the heart muscle won't wear out, won't overexert itself to the point where it can no longer contract. And the final muscle type that we want to talk about is what we call smooth muscle. So you can see smooth muscle here embedded in the walls of organs, where it enables internal organs to move stuff through them in a rhythmic fashion. For instance, it's in the lining of the stomach and the esophagus and the small and large intestine, and it also is in the lining of the blood vessels and the lymphatic vessels, and it's also attached to every hair in your body. It's under involuntary control. It's not striated, and the muscle cells in smooth muscle are shaped like a spindle, meaning they're fat in the middle, and they tend to be tapered on the ends. There's one nucleus per cell, and there are no visible striations, and it is not under voluntary control, meaning we don't tell it what to do. You can also see connective tissue between the muscle cells that hold the smooth muscle together so that it doesn't come apart when it contracts. And this is primarily endomysium. And the speed of contraction is very slow in smooth muscle because it doesn't need to be quick in order for it to do its job. It is capable of rhythmic contractions known as peristalsis, which are the waves that move through, for instance, the digestive system that help propel food through the digestive tract so that it ultimately goes from the oral cavity to the esophagus to the stomach, and from the stomach to the small intestine, and from the small intestine to the large intestine. And that's only possible because this tube is capable of squeezing, and when it squeezes, it pushes its contents through the tube in a very regular manner, okay, stimulated by the presence of food in the tube. Other places where you've seen smooth muscle at work include when you get goosebumps in response to cold temperatures or fear and your hair stands up on end. That's the action of smooth muscle that's attached to the hair follicles that causes them to create that effect. Other places where smooth muscle acts are in the pupils of your eyes, okay? When your pupils dilate or constrict, it's smooth muscle activity that is responsible for that as well, okay? So smooth muscle does a lot of important things throughout the body. What we've talked about mostly in this exercise and in lecture is this muscle type, okay? Skeletal muscle, which makes up most of the muscles that we've discussed in this chapter. Okay, examples of skeletal muscle include the biceps femoris, the gastrocnemius, the deltoid muscle, and so on. And remember that this is the only muscle type that we control with our conscious thought. The other two, again, are on autopilot. Okay, another thing that we want to address when we talk about muscle are a couple of features that we need to be aware of when we're trying to describe a muscle's location and its job are the origin, the insertion, and the action, okay? So very important to understand the difference between these three, okay? So if we look here at the origin, okay, what we're talking about when we talk about the origin is the muscle's attachment to immovable bone, okay? So the muscle has its start here, and then it'll cross a joint. In this case, it's crossing the elbow joint, and then the insertion, okay, is its attachment to movable bone. And so this muscle is the brachialis. It has its origin on the diapasis of the humerus, and it has its insertion on the diapasis of the ulna, and so it crosses the elbow joint. And when the muscle shortens, okay, so let's visualize the muscle shortening. Okay, when this muscle gets short, what happens is that we have elbow flexion, right? You can see the elbow here going up, okay? Elbow flexion. So origin, insertion, and action, okay? Flexion meaning to reduce the angle between the two bones, right, as a result of the muscle getting shorter. Generally, muscles are attached to bone by connective tissue known as tendons, and as a result, they're able to translate their force from the muscle fiber directly to the bone. If we zoom in on the organization of muscle, we see some interesting features. First of all, we see three layers of connective tissue that are important in holding the muscle together so that when it contracts, it doesn't come apart. The outer layer here is called the paramecium. Okay, you can see the epimecium. You can see the epimecium here surrounding the very outside part of the muscle. Or surrounding each fascicle is the paramecium, okay, around these bundles of muscle cells. These bundles of muscle cells are called fascicles. And then surrounding each individual muscle fiber is endomecium. Okay, so you got three layers of connective tissue that help to hold the muscle together so that it doesn't come apart when it contracts. In addition, you've got connections of the epimecium directly to the periosteum of the bone. Remember that that's the outer membrane of bone that contains living cells that help build bone and also blood vessels that help nourish bone, all embedded in connective tissue. And this attachment is what allows the force of the muscle to be translated to the bone itself. Okay, so you can see here the tendon and the periosteum around the outside of the bone, and then of course how the connective tissue holds the other muscle components together. So it's a little bit, can be a little bit confusing, but if we go from the inside out, looking at a muscle, let's go with the very smallest thing. The very smallest thing is something called a myofibril. And what is a myofibril? It is a muscle protein that's inside the muscle cell. Okay, so you could think of it as a long band of protein that runs the entire length of the muscle cell from end to end, and these myofibrils are stacked one on top of the other so that it looks like basically a stack of firewood inside the cell, okay, kind of all held together by the cell membrane and to some degree the organelles inside the cell. And so what these muscle proteins do is they're capable of contraction, they're capable of shortening when they use the energy source ATP. And so as the myofibrils contract, the myofibers, okay, the muscle cells contract. As the myofibers contract, the fascicle will contract, and as the fascicle contracts, the entire muscle will contract. Okay, so that's how the muscle is able to produce movement, by its ability to shorten itself. Okay, so always remember, muscles produce force by contraction. So what we want to do now is talk a little bit about exactly how this contraction happens. Okay, and the reason that this can occur is because of the unique arrangement of the proteins inside a muscle cell. So what you're looking at here is just a little piece of a single muscle cell. Notice that you have more than one nucleus in skeletal muscle, and you have lots and lots of protein. All these little logs here that look like little red logs, those are all collections of muscle protein, and they're bound together by the cell membrane, and also to some extent the cell endoplasmic reticulum, and of course the individual muscle cells are held together by connective tissue on the outside. So how does a muscle contract? So one of the things we want to do is we want to look at the organization of a muscle cell. Each muscle cell has an outer membrane that has a special name. It's called the sarcolemma, and many nuclei that are there to direct the generation of muscle protein. They express the instructions for myosin and actin and the other components of muscle protein to a great degree. Okay, they read from the muscle handbook of DNA all the time, so they're constantly making new protein and repairing the protein that they have. Within the cell are myofibrils, which are made up of actin and myosin filaments. The myosin filaments are a bit thicker than the actin filaments are, and they have a golf club shaped head to them that's able to grab onto the actin and pull it using ATP as an energy source. And so you could think of it working in a way a little bit similar to how it is that a jack can jack up a car. Okay, if you've ever had to jack up a car with the need to change a tire, and you've noticed how the jack works, it's able to raise the car off the ground because each time you pump the jack will crawl up the bar that elevates the car one notch at a time. Okay, and the energy to do that is actually coming from you, the force of your muscles. And the same is true really here with the arrangement of proteins in the myofibril. You could think of them as a series of jacks that crawl over the top of each other, causing the myofibril to shorten. The A bands are the dark bands, and the I bands are the light band, and they are indicative of the overlap of the proteins in these fibers. Basically, when the fibers overlap, they block light from passing through them, so that's why they look dark under a microscope. And if we take a single myofibril and blow it up so that we can see it, we start to see a little bit of how the muscle proteins are arranged. So this is a drawing of what the muscle proteins look like, and you can see here the thin filaments, which are shown here in blue. Those are the actin. The thick filaments here shown in red, that's the myosin. And then you've got anchoring proteins that sort of hold the myosin into place, and it's these myosin fibers that reach out and grab the actin and pull on it when the conditions are right. Okay, so the actin filaments are pulled by the myosin filaments, and what they do is they migrate towards what we call the M line. This is the M line here. There's an M line, there's an M line, and there's an M line. And you could think of the M lines as an area inside the muscle protein where the the myosin is sort of stapled into place. It can't move. All it can do is reach out and pull on stuff that's on either side of it. The Z discs are areas of attachment for the actin filaments, and they contain other anchoring proteins to which the actin can attach. And so when the muscle shortens, what happens is, and we'll draw it here, that the Z discs will move towards each other, okay, like this, okay, as the muscle fiber contracts, as these myosin filaments, the red things here, reach up and pull on the actin and yank these Z discs closer to each other. And this happens all up and down the fiber, and this is why the fiber gets short when the muscle is engaging in a contraction. Other features of the muscle myofibril we want to be aware of, the M line, the I band, which is the light area, the area of non-overlap between actin and myosin, the A band, which is the area of overlap, and the H zone, which is the area where neither the actin or the myosin overlap to any great degree. It's basically a point of attachment for the myosin, and so this is one of the constants in the sarcomere as the muscle contracts and the fibers walk over the top of each other. And the distance from one I band to another I band, or from Z disc to Z disc, if you like, is what we call a sarcomere. Okay, that is the contractile unit of skeletal muscle, and you've got hundreds of sarcomeres that are arranged end to end to end to end to end, going all up and down the fiber. So you can see what happens when the sarcomeres start to shorten is that the fiber will shorten considerably as well. And the energy source for this, again, is ATP, the universal energy currency that's used by cells to do work. ATP, right there, okay? All right, that's what the myosin uses to grab and pull. That's the gas in the tank that drives this entire process. And so here you can just see how it is that the myosin and the actin interact. This is the muscle at rest. These are the actin filaments here. This is the H zone, okay? And in the middle here you can see the M line. This is the myosin, okay? The thick filaments of myosin that at rest can't reach out and grab the actin because it's blocked. And then when the muscle decides that it wants to contract, when it gets an electrical signal saying produce a force, okay, from the nervous system, what happens is that a series of chemical reactions inside the cell cause these actin filaments here to pivot a little bit so that they reveal a place on the actin filament where the myosin can reach up and grab, and this is what we call cross-bridging. And so what happens is that we make a connection between the actin and the myosin. That allows the myosin to pull on the actin, and you can see what happens is that the Z discs get closer together as the myosin pulls on the actin and shortens the entire sarcomere. Okay, so this again all requires ATP to work. This is just a zoom up showing you a little bit more detail of how the myosin and the actin interact. At rest, before the contraction begins, the myosin head next to the actin is close by but is not physically attached to the actin yet. It hasn't reached out and grabbed it in order to pull it and make the muscle shorten. And so what it's waiting for is a series of events that will allow these binding sites, these little handles on the actin, to be available for the myosin to reach up and grab. And that event that causes that to happen inside the muscle cell is the release of calcium from the endoplasmic reticulum of the muscle cell, which has a special name. It's called the sarcoplasmic reticulum. And what the calcium does is it binds to the actin and causes it to pivot so that the myosin can reach up and grab the actin, and then along with ATP as an energy source start to pull on the actin and shorten the sarcomere so that everything gets small, essentially. Okay. So what you're looking at here is just the calcium ions entering the cytoplasm of the muscle cell and binding to the actin, and this causes the actin filaments to pivot so that the myosin binding sites are available for the myosin to reach out and grab. So you can see here the calcium flooding inside the cell, binding to a site on the actin that's called it's a protein. It's called troponin C, and this is hooked to the actin filament, and this causes it to pivot so that the myosin can reach out and grab. What happens next is that the myosin heads will pivot. Okay, as the calcium ions become available, the heads will move after they've bound, and they'll pivot, and then they'll let go and they'll recock. The free myosin heads use the energy from ATP to orient themselves in an active shape, okay, a shape that contains power, like a mousetrap being set um, with the retainer that holds back the spring. The physical movement of the attachment of myosin to actin is what springs the trap, and this causes the myosin heads to snap towards the center of the sarcomere. Because the actin and the myosin are bound to each other, when this happens, the thin filaments will be pulled towards the center of the sarcomere, and the entire sarcomere will shorten. The ATP is where the energy comes from, and it is needed to both release and recock each myosin head so that it's ready to reach out and grab another myosin binding site on the actin, as long as there's calcium around as a result of the electrical signal coming from the motor neuron that's telling the muscle to contract. So it's a series of events, right? The the nerve cell that's going to tell the muscle to contract sends an electrical signal. That electrical signal um, goes inside the muscle cell, and that causes the release of calcium. The calcium then binds to the troponin that's on the actin, that causes the actin to pivot, and then the myosin reaches up and will grab the myosin binding sites on the actin, and then using the energy from ATP will pull on these actin filaments and basically walk over the top of them, causing the entire sarcomere to get shorter. So you can see that going on here, right? The pivoting of the actin heads as they pull these actin filaments closer to the midline of the sarcomere. When the action potential ends and calcium ions are reabsorbed back into the endoplasmic reticulum of the muscle cell, called the sarcoplasmic reticulum, uh, the regulatory proteins go back to their original shape and position, and they block the myosin binding sites from the thick filaments, and as a result, the myosin can't make cross bridges anymore, and now, since it has nothing to attach to, the muscle cell will relax and go back to its original length. And what you're looking at here is just an explanation of where the energy comes from to provide all of this contractile force. So initially, the energy comes from a special molecule inside the muscle cell called creatine phosphate. It's the phosphate bond between the phosphate and the creatine that contains the chemical potential energy, which can be used to make a molecule called ATP, which is what the myosin heads on the myosin protein are going to use as an energy source. You get one ATP molecule for every creatine phosphate that's used to extract energy, and the duration of the energy is about 15 seconds per molecule. Say this is very rapidly used up. Now, where else does the energy for muscle contraction come from? And it comes from the chemical bonds in the food that we eat. There's energy contained in the attachments of atoms to other atoms, and when you cut those attachments open, the energy that comes out can be trapped and used to make other molecules that the cell can immediately use to do work. In aerobic respiration, which is the kind of energy production that occurs when there's enough oxygen around, what happens is that the most prevalent fuel that muscle will use is glucose, which is available in two places. It's available inside the muscle cell itself as a polymer called glycogen, and it's also available from the bloodstream as free glucose molecules. And what will happen when the muscle needs energy from glucose is that it will transport it inside the cell using special pumps that are on the muscle cell membrane that will pump the glucose from the blood into the cell cytoplasm. And then what will happen next is that the chemical bonds in glucose will slowly be split apart. And when they're split apart, that energy will be used to make the molecule that you can think of as what I like to call biological gas in the tank, which is ATP. ATP stands for adenosine triphosphate, and it is the form in which energy is most easily used by living systems. So what happens when glucose enters the cell is that it's broken apart from a 6-carbon molecule into 2 3-carbon molecules, and along the way what happens is that a small amount of ATP is generated in that process. The next thing that happens is that 3-carbon molecule that came from the initial set of chemical reactions that happened in the cell cytoplasm, the pyruvic acid, shown here in this diagram by the little box, that will enter the powerhouse of the cell known as the mitochondria. And inside the mitochondria, what will happen is that the chemical bonds in the pyruvic acid will slowly be broken apart, and the energy from those chemical bonds will be used to create a whole lot more ATP that the cell can use in order to perform work. For every glucose molecule that enters this set of pathways, which this pathway is called glycolysis up here, and the pathway that goes from pyruvic acid all the way to carbon dioxide, water, and ATP is actually 2 chemical pathways that occur in the mitochondria. One is called the citric acid cycle or Krebs cycle, and the other is called the electron transport chain. What they do together inside the mitochondria is make a whole lot of ATP that the cell can use for work. So for every glucose molecule that goes through these 3 pathways, you get between 36 and 38 ATP per molecule of glucose. And that's what's happening inside the muscle cell itself and inside the mitochondria. And that's where the ATP comes from in order to drive muscle contraction for a long period of time. Now, if we don't have oxygen around, we have a problem, which is that we can't run the chemical pathways that occur inside the mitochondria here. And so, as a result, what will happen is that glucose will be used as an energy source, and some ATP will be produced from that glucose, but the pathway basically stops at pyruvic acid, and that pyruvic acid will be converted into lactic acid inside the muscle and eventually transported away in the bloodstream where it'll reach the liver, and the liver will then convert it back to pyruvate and hopefully generate enough energy so that we can continue to fuel our activity. But what happens inside the muscle cell is that you're making only 2 ATP per molecule of glucose that enters this very short chemical pathway, and that's not enough really to perform activity for a very long period of time. This is what we call anaerobic respiration or anaerobic glycolysis. The formation of the lactic acid actually ends up damaging the muscle tissue if it goes on for very long, and what will eventually happen is that the muscle cell will run out of energy, and the accumulation of lactic acid will damage the muscle fiber to the point where the activity can't continue. And at that point, the muscle will fail, and so the activity will stop. So 3 different ways that the cell is getting energy here, directly as a result of its creatine phosphate stores transferring the energy right to ATP. When you've got adequate oxygen supply, you're using both glycolysis here and the Krebs cycle and the electron transport chain inside the mitochondria to make a whole bunch of ATP. And then without oxygen, if we're more active than we have oxygen to supply our metabolic needs, what happens is that we only make 2 ATP per molecule of glucose, and eventually the activity will stop. Okay, what you're looking at here is just a chart showing you the different types of metabolic pathways that are used in different types of activity regimens. When we're doing long distance running, okay, an activity that goes on for a long time, but that allows us to take in enough oxygen to sustain the activity, we use all three pathways: glycolysis, the Krebs cycle, and the electron transport chain. Whereas when we're doing something short term, often we only need a little bit of energy very quickly, and so we use creatine phosphate initially, and then we can generate the rest of the ATP for the activity simply by using glycolysis, which is going to generate some ATP and is going to start to build up lactate in the muscle. And this will only last about a minute and a half or so, whereas this can go on for hours and hours. Now, another thing we want to understand is how it is that the muscle gets the signal to contract in the first place. And that comes from another type of cell called a nerve cell. Okay, a nerve cell, sometimes called a neuron, is a cell that is specifically designed to send and receive electrical signals so that it can tell target tissues, in this case muscle, to do something. So what you're looking at here is an example of how it is that we decide to have a skeletal muscle contract. First, we make a conscious decision to do something, to perform some sort of movement. And that information will go from the brain to the brain stem, and then it's going to go down through a long tube called the spinal cord that runs inside your vertebral column. And when it gets to a level of the vertebral column that's fairly close to the muscle that the nervous system wants to talk to, the signal will leave. This is the spinal cord, and it will head out through a motor neuron that'll go all the way from the spinal cord to the skeletal muscle fibers. And so this is one long cytoplasmic process that comes out from the nerve cell, which is called the axon, and this is what carries the electrical signal all the way to the muscle cell. And what will happen then is that it'll stop at the end of the axon, which is called the axon terminal. And at that point, what will happen is that the membrane that covers the axon terminal will release neurotransmitters, which are special chemicals that will float across the tiny gap that exists between the muscle cell and the nerve cell, which is called the neuromuscular junction, and then it will bind to receptors that are on the muscle cell membrane, and that will cause electrical changes in the muscle cell membrane that will eventually cause the muscle to contract. And we've got some really good videos that I'll show you at the end of this podcast that illustrate each of those phenomena happening. You can see here the neurons that lead out to the muscle cell fibers, and you can see the muscle cell fibers themselves, and you can see also the neuromuscular junctions here and here and here and here and here. Now, one of the things that we need to be aware of is the fact that different motor neurons are going to talk to different muscle fibers. And so when a nerve heads out to a muscle, what will happen is that a whole lot of different nerve axons will touch a whole lot of different muscle fibers, and the strength of the contraction will in part be due to how many of these motor neurons fire, and as a result how many of these muscle fibers are told to contract. So if both of these, what we call motor units, were to fire together, you would make a much stronger contraction than if one or the other were to fire separately. Okay. The other thing that increases the force of contraction has to do with how frequently the nerve cell fires telling the muscle to contract. If the nerve cell fires very rapid in succession, like you're going to make a much stronger contraction than if the nerve cell were to fire at a much slower rate, like dit, dit, dit. And as a result, both of these factors can influence the strength of a muscle contraction as well as its duration. So it's the frequency of how often the nerve fires and also the number of neurons that fire that indicate how strong the contraction is going to be. And this is just a piece of tissue taken from a neuromuscular junction, and what you can see here are the branching axons of the motor units, and these little dark areas are the motor end plates, and they sit right on top of the neuromuscular junction, and that's where the neurotransmitter is released that tells the muscle fiber to contract. And so what you're looking at here is how the signal moves um, from one place to another along the muscle cell. So this is the neuromuscular junction. The release of the neurotransmitter causes a change in the voltage on the membrane around just this particular area, and then what happens is that leads to a series of electrical changes that move both along the muscle fiber and deep inside the muscle fiber as it spreads from where it starts all the way to where it ends in sort of an unstoppable cascade of events, just like tipping over the first domino in a line of dominoes. This signal will spread all the way to the ends of the muscle fiber. And so you can see here that what will happen next if we go inside this neuromuscular junction and look at a little bit more detail is that as the electrical signal again reaches the end of the axon, what happens at the microscopic level is that calcium gates will open up, and the calcium that lies outside the nerve cell will rush inside the nerve cell, and that will trigger the fusion of these synaptic vesicles, which are little bubbles of membrane that contain the neurotransmitter, and that will make them fuse with the axon terminal. This membrane here is called the presynaptic membrane, and that will cause the neurotransmitter to be released into the neuromuscular junction. And that'll happen next. If you look down here as a zoom up, you can see that the neurotransmitter, which in this case is acetylcholine, binds to these little channels, and when those channels open up, it allows sodium to move inside the muscle cell and potassium to move out. And what will happen is that you'll get what we call a change in membrane potential at that point, and the result will be what we call a depolarization, and that's what suffices as an electrical signal in living tissue. And as that depolarization spreads from where it starts all the way inside and along the muscle fiber, those electrical changes get carried deep inside the muscle cell through little channels that are called T-tubules that move all, they're sort of deep tunnels that start at the membrane surface and go deep inside the muscle cell, and they carry the electrical signal all along the sarcomere, which is covered by the endoplasmic reticulum of the muscle cell, which is called the sarcoplasmic reticulum. And as a result, what happens is that the calcium that's inside the sarcoplasmic reticulum, inside the muscle cell, gets released. You can see that here. And the binding of that calcium to these special sites on the actin filaments of the sarcomere causes those actin filaments to pivot. As those actin filaments pivot, the myosin heads reach up and grab them, and they start pulling on the actin, and that's what's going to cause the muscle to shorten. Okay, and what I'll like you to do is to watch the videos that I have right after this podcast. It'll go into a little bit more detail how this works. It'll probably give you a little bit more information than you need to know, but I think it's nice to see it illustrated versus just having me talk about it and indicate what's happening on these different diagrams. So we end up with the question of, what sort of muscle tissue predominates your physique, and depending on your activity regimen, we can either be a couch potato or an endurance athlete or a weightlifter or a sprinter, and that's going to dictate the type of muscle tissue that you're going to find most frequently inside the body. Essentially, if you're a long distance athlete, you're going to have a lot of what we call red meat, okay, or dark meat. You'll have muscle tissue that has a high oxygen content, so it has a lot of a molecule called myoglobin in it, and it's not going to have tremendous amounts of sugar stored inside the muscle because it'll get most of its nourishment from the bloodstream. Whereas if you're a weightlifter or a sprinter, your muscles will tend to be what we call white meat. Just think of a bucket of chicken here. The muscle tissue tends to have a lot of sugar stored in it, relatively little myoglobin, and it fatigues very quickly as compared to the muscle tissue that you would see most often in a long distance runner. And then, of course, if you're a couch potato, your muscle is going to be have a very low energy content comparatively. You're going to both be incapable of endurance activity as well as short bursts of sustained activity because you haven't stimulated the muscle tissue enough via the nervous system to remind the muscle tissue that it needs to keep making muscle protein, and the result is that your muscle mass and your muscle strength will decline over time. This is why exercising makes us stronger, because it causes the nervous system to tell the muscle tissue to make muscle protein, and that's what causes the muscles to get bigger. So this is something we want to keep in mind when we're talking about exercising. Okay, stay tuned for some nice videos on the neuromuscular junction and excitation-contraction coupling that'll explain this in a little bit more detail, and I'll see everybody in lab.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine. Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers. The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released. and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.